Hello and warm welcome, students. Welcome to Global Academy of Medical Education. Today's topic, it is such a topic that if at least one topic you need to cover in microbiology, you cannot miss rickettsial infections. Because over the year, every year, there has been more than one questions that has been asked from rickettsial infection. So, when we see this image, we see some arthropod vectors. So there are ticks, hard and soft ticks, then there are mites, trombiculid mites, and we have flea and louse. So flea and louse, these are also vectors for various rickettsial infections. Now what are rickettsial infections? Some general properties about them. So they are small, non-motile, gram-negative coccobacilli, and the characteristic feature is that they are obligate intracellular organisms. They are obligate intracellular. So any of the previously there were some bacteria that were not obligate intracellular but still classified under Rickettsiae, but now they have been excluded, for example Coxiella and Bartonella. So another feature they have is not cultivable in artificial media, and they grow in cell line by animal or egg inoculation. So this is an important property because Bartonella, it can be grown in blood agar. So now they are not included in Rickettsia, and all of them they are transmitted by arthropod vectors such as tick, mite, flea or louse. And their final target site, it is vascular endothelial cells. This is the final target site. Now classification. In the order Rickettsialis, we have two family. One is Ricketsiaceae, another is Anaplasmatidae. In Ricketsiaceae, we can classify it into two genus, Rickettsia and Orientia. In Anaplasmatidae, we have four genus. We have Ehrlichia, Wolbachia, Anaplasma and Neorickettia. So again I have classified the same. In family Rickettsia, we have Rickettsiae, and Ricketsiaceae we have Rickettsiae and Orientia, and in family Anaplasmatidae we have Ehrlichia, Wolbachia, Anaplasma and Neorickettia. So like I said, the former members such as Coxiella and Bartonella have been excluded from the study, from the family, although in today's topic of discussion we are going to know some characteristics about them also, but they are excluded from the family Rickettsia because Coxiella is not arthropod borne, it is transmitted by inhalational mode, and Bartonella is not an obligate intracellular parasite, it can be grown in cell-free media. So therefore they are excluded. Now seeing the characteristics of Rickettsiae, like their small size and their obligate intracellular properties, it was initially thought that they were viruses. But now they are confirmed to be bacteria because they possess a gram-negative cell wall and both DNA and RNA. Although the staining is better with GIMSA stain, but they are gram-negative. They can be also stained by gram stain, and their gram-negative cell wall contains lipopolysaccharide peptidoglycan. They multiply by binary fission. They are susceptible to antibacterial agents, and they are large enough to be seen under the light microscope and held back by bacterial filters. So all these properties differentiate them from viruses. So history regarding Rickettsia is also very important because Howard Taylor Ricketts and von Provozic, on Howard Taylor Ricketts, who discovered Rocky Mountain spotted fever, which is transmitted by a tick, and von Provozic, who both of them, von Provozic and Howard Ricketts, they died of typhus fever, which they contracted during their study. So giving tribute to their contribution, the genus Rickettsia has been named after the discoverers. Now, family Rickettsia, this is again, we have repeated that they consist of two pathogenic genera, Rickettsia and Orientia. So, infections can be categorized into two broad groups. One is the typhus fever group and is the spotted fever group. Coming to the antigenic structure, cell wall, I have told you that it is similar to that of gram-negative bacteria. It consists of lipopolysaccharide, peptidoglycan and some outer membrane protein antigens. Some outer membrane protein antigens, which are species specific. They are highly immunogenic and therefore they can be used for both vaccine as well as for serodiagnosis. So this is important. Outer membrane protein antigen, since they are highly immunogenic, they can be used for vaccine as well as for serodiagnosis. Next we have is the lipopolysaccharide antigen, are non-group specific and they are found in some Rickettsia, and this is shared by certain strains of Proteus. So this forms the historical basis of the Weil-Felix reaction, which is based on the presence of heterophile antigens, some shared antigens between Rickettsia and some non-motile strains of Proteus vulgaris, OX19 and OX-K of Proteus mirabilis. Coming to the pathogenesis, by now we know that the transmission is by arthropod vectors and spread is via lymphatics from the portal of entry. They multiply in the regional lymph node and then spread via bloodstream. And the target cell, these are the endothelial cells. So these outer membrane protein and A and B, they adhere to the endothelial cells, and following adhesion the organisms are phagocytosed and they resist the lysosomal killing or degradation by inhibiting the phagolysosome fusion, after which the bacteria, they multiply by binary fission. Generation time is 9 to 12 hours and it is spread from one cell to another by actin polymerization. So this is in brief about the pathogenesis. Now this broad table, this is the summary and it will help us in diagnosing majority of the infections of the like I said, like Rickettsia, the infections can be broadly classified into the typhus group and the spotted fever group. Now in typhus group, what are the species we have? So the important species we have is Rickettsial prowazekii and Rickettsial typhi. Now how to remember? In Rickettsial prowazekii, both like in the typhus group, we have epidemic typhus and we have endemic typhus. So from the typhus you can remember that the disease will be epidemic or endemic. Now how to remember that Rickettsial prowazekii causes epidemic typhus? We can see that Rickettsial prowazekii and epidemic, you can remember the P. So from that you can write the choose the answer that epidemic typhus is caused by Rickettsial prowazekii and the chronic recrudescent form is known as Brill-Zinsser disease, which is seen after many years, and it is due to when the immunity wanes off, it is again recrudescence of the illness. Now vector is louse. And it is seen worldwide. And what is the characteristic of the rash? The characteristic of the rash is it is all over the body except the palm and sole. So extremities are spared. And what is eschar? Eschar is the point of entry of the vector where there is a necrosis and surrounding there is hyperemia. So that is absent. There is no eschar, there is no lymph node enlargement. And in Weil-Felix reaction, you have to remember like OX19 and OX2 OX19 and OX2, it is like from the non-motile strains of Proteus vulgaris. Non-motile strains of Proteus vulgaris. This is the antigen, and we add the patient's serum in dilution, and it is a tube agglutination where we see that the OX19 is strongly agglutinated and OX2 may be plus minus. This is in case of epidemic typhus. So in case of Brill-Zinsner disease, it is after several years of infection by Rickettsial prowazekii epidemic typhus, the patient may again be presenting with features of this fever, typhus fever, epidemic typhus. But in this case, since antibody has fallen after due to waning immunity, it is negative Weil Felix or weakly positive. So in the typhus group, we have discussed about Rickettsial prowazekii, epidemic typhus and Brill-Zinsner disease. Now about Rickettsial typhi, it is again the second variety of the typhus group. So here to remember, I will give you the mnemonic ENT from endemic and typhi. So Rickettsial typhi, it is spread by the vector is flea and worldwide distribution, and here also the rashes are there in the mostly in the trunk and sparing the extremities. What about the Weil Felix reaction? It has got similar pattern. OX19 will be strongly reactive and OX2 will be either reactive or non-reactive. So this was about the typhus group. Let us come to the spotted fever group. So in spotted fever group, we have first is Rickettsial rickettsii. Now to remember the disease caused by Rickettsial rickettsii, we have all known that the movie RRR has become so popular. So Rickettsial rickettsii, you can remember like that, Rickettsial rickettsii and Rocky Mountain spotted fever. So this is the mnemonic for the same. Name itself spotted fever group, you will see that all of them are vector is tick. In case of typhus group, epidemic typhus was spread by louse, endemic typhus by flea, and here all the Rickettsial spotted fever group except the pox is spread by vector is tick. So Rocky Mountain spotted fever, it is uncommon in India. It is mostly seen in the US, and the rash, see the difference. The rash is 90% in the extremities and less in the trunk. So it can be differentiated from the typhus group. And eschar is also less. And Weil Felix reaction, we will find that both OX19 and OX2 are positive. So both there will be slide agglutination, tube agglutination with both OX19 and OX2 when we give the non-motile strains of Proteus vulgaris. We will see that there is agglutination taking place. Now in the spotted fever group, we have the second variety as Rickettsial conuri. Rickettsial conuri, it is the disease produced is Indian tick typhus. So I will put the mnemonic I, T and C. ITC, which is a popular chain. So ITC, Rickettsial conuri, it is found in India. It is there in Asia. Here also there is rash and there may be eschar also seen. Eschar is seen at the site of entry of the vector. And lymph node swelling will be there. And both OX19 and OX2 will be agglutinated. In Rickettsial Africa, it is African tick bite fever seen in sub-Saharan region and not very common in our region. And here the eschar is most prominently seen 90%, and also another important clinical feature is that the lymph nodes will be highly enlarged, lymphadenopathy. The last group we have is Rickettsial pox. Now Rickettsial pox, this from the pox it is sounding similar to chicken pox. So because this has also got vesicular lesions, and it is the species implicated is Rickettsial acari, and here the vector is different. It is not tick, it is gamma seed mite. And here the rashes entirely it is vesicular, and eschar is very prominently seen. Lymphadenopathy is seen, and in wheel felix reaction we do not find any reaction, all negative. Then in Rickettsiaceae we had classified into Rickettsia genus and into Aurantia. So the third broad group we have is scrub typhus. The species is Aurantia shushugamushi. The disease caused is scrub typhus. Here also the vector is mite, trombiculide mite, where the larval form, the Shiggers, is responsible. Larval form Shiggers is predominantly responsible. Distribution it is seen in Asia, Australia, common in our region, in Indian country, in India. And here rash and eschar, rash is also seen 50% of the cases, eschar is seen in 35% or sometimes missed. Lymphadenopathy is seen, and wheel felix reaction it will be ox K will give strongly positive, which is non-motile strains of Proteus mirabilis. Now let us take one at a time. Most of the points have been covered in the broad classification that we have made, but yet we are going to repeat it because these are often asked questions. So epidemic typhus, again we know that it is louse borne. It is seen in closed conditions where there is lot of people in unhygienic condition. So it is caused by Rickettsia prowazekii. I told you the mnemonic for here is P and P, Rickettsia prowazekii, causing epidemic typhus. And vector is human body louse, Pediculus humanus corporis, acquires the organism while taking the blood meal from an infected patient. Here the rashes are in the trunks. It spares the face and the extremities. So mode of transmission by rubbing or scratching of abraded skin or mucosa contaminated by louse feces. Rarely it may be due to inhalation of louse feces during laboratory work exposure, or it may be also used as an agent of bioterrorism. Now epidemic typhus, louse borne, the clinical manifestations. So it is a febrile disease, acute febrile disease accompanied by headache, myalgia, and eye discharge and rashes after an incubation period of 1 to 2 weeks. Now characteristic of the rash has already been discussed. It is more in the trunks, leaving the extremities. Myalgia. Now myalgia, it has got a severe myalgia, like there is a in the epidemic typhus there is an outbreak that happened in Burundi region, and there the patient will be crooked, trying to seek respite from the pain. So the myalgia is very severe, and complications it can lead to is interstitial pneumonitis, CNS manifestations as mental confusion and coma. So here I will like to mention is that the term typhus, in actually it means the comatose state. It is the drowsy state or the comatose state that a patient results, and that from that the nomenclature of typhus has been derived. So whenever there is when the louse population increases in unhygienic condition, it is a very prominent risk factor. Zoonotic cycle is maintained by eastern flying squirrels, and the lice and fleas maintain Rickettsia prowazekii in the environment. And endemic in Africa and South America, where Burundi outbreak in 1997 involved nearly 1 lakh people in refugee camps. So this was the outbreak I have previously spoken about. Now what is Brill-Zinsser disease? It is the recrudescence illness occurring years after acute epidemic typhus. The Rickettsia prowazekii, it remains latent for years, and reactivation occurs due to waning immunity, which leads to sporadic infection or outbreaks. So that was all about epidemic typhus. Now we have endemic typhus. So I told you the mnemonic to remember was ENT, endemic typhus. So endemic murine typhus, it is caused by Rickettsia typhi infection. Here the vector is flea, rat flea, or cat flea. Rat flea is the scientific name is Xenopsylla chiopsis, and cat flea is Tenocephalidis felis. Mode of transmission is rubbing or scratching on skin or inhalation of fleas, dry species, less frequently by flea bite. And what is the reservoir? The reservoir is rodents, which is Rattus rattus or Rattus norvegicus. Now coming to endemic manifestations of endemic typhus, it is a milder form of illness similar to epidemic typhus, but here it is milder and rarely fatal. And the rash, apart from fever, headache, myalgia, anorexia, the rash involves, again, the I told you, in all the typhus fever group, the rash will be concentrated in the trunk and than the extremities. It is endemic worldwide, especially in warm tropics having high rat infestations. In India, Shimla, Kashmir, Mumbai, Jabalpur, Lucknow, Pune, all these have reported cases. Now we are coming to the spotted fever group. Now in the spotted fever group, the first group was Rocky Mountain spotted fever, and here also the mnemonic is RRR. Rocky Mountain spotted fever caused by Rickettsia rickettsii. And all the spotted fever group I told you was big vector was tick, except Rickettsial acari, which causes Rickettsial Box, that is transmitted by Gamseed mites. Here the vector is tick. In USA it is and we are fortunate that this does not occur in our country. It is predominantly it is seen in the US, and the vector is Dermacentor andersoni in US and Ambyloma caginense in Central and South America. You can remember Dermacentor and Ambyloma. And transmission is by bite of ticks. Reservoir, the ticks serve as vector as well as reservoir, and other mammals like dogs and small rodents may also act as reservoir. Now manifestation. So the spotted fever group, the contrast with the typhus fever group is that the rashes occur on the extremities, that is the wrist and the ankle and the trunk. Initially they may be maculopapular but later become hemorrhagic. And incubation period 4 to 14 days. Of course there will be fever, there will be headache, there will be rash and frequent myalgia and anorexia. So these are common but characteristic rashes we can differentiate. Complications, they appear late, and it may be disseminated intravascular coagulation, interstitial pneumonitis, CNS involvement, and myocarditis and renal failure. It is the most fatal rickettsial disease. So this you have to, we can note that most fatal rickettsial disease is Rocky Mountain spotted fever. Geographically, I told you there is high tick population in the US, Central and South America, so it's distributed mainly in that region and mostly seen in summer in children and males. Coming to the Indian scenario, we have Indian tick typhus. Here I am repeating the mnemonic we used is ITC from Indian tick and conuri. Indian tick typhus caused by Rickettsial conuri. It is transmitted by again a tick bite, which is Rhipicephalus sanguinis. Clinical manifestation, we have a eschar that occurs at the site of the tick bite. Eschar is the deep necrotic region surrounded by a hyperemic region. We are going to see an image of it, and it is prevalent in southern Europe, Africa and southern Asia. So this we have a very prominent image of an eschar. We see that there is a black necrosis and which is surrounded by a hyperemic region, red hyperemic region. Now Indian tick typhus in India, it is from the name suggestive, it is widespread, seen in Nagpur, Jabalpur, Sagar, Pune, Lucknow, Bengaluru, Secunderabad. Now coming to Rickettsial pox. Rickettsial pox, similar to the rashes are similar to chicken pox, and therefore the same vesicular lesions, and they're confusing the diagnosis. It is caused by Rickettsial acari, and it is transmitted by all the spotted fever groups were transmitted by ticks except the Rickettsial pox, which is by mite. Liponisoides sanguinis. Reservoir here we have is mice, which is the principal reservoir of Rickettsia acari. Manifestations similar to other rickettsial disease but differ from the latter by the presence of vesicular rashes. There in other spotted fever group we had maculopapular rashes. Here we have vesicular rashes. We have a eschar prominent and regional lymphadenopathy. So it is again endemic in US, Ukraine, Turkey and Mexico. Now coming to the diagnosis of rickettsial infection, rickettsioses, while discussing the antigenic structure and also in the broad classification, we have learned about the Weil-Felix test. So Weil-Felix test is a non-specific test based on the concept of heterophile antigen. So this is a shared antigen between Rickettsia and the non-motile strains of Proteus. Which are the non-motile strains of Proteus? OX19, OX2 and OXK. OX19 and OXK, OX2 of Proteus vulgaris and OXK of Proteus mirabilis. In epidemic and endemic typhus, if we go back and look at the chart, I have told you that there is only rise in the OX19 antibody. In tick-borne spotted fever, which is the Rocky Mountain spotted fever, the Indian tick typhus, the Rickettsia africa, we have both increased OX19 and OX2 antibodies, and in scrub typhus, we have increased OXK antibody. So this was about the Weil-Felix test. Now specific antibody detection, it is by indirect immunofluorescence, ELISA, enzyme-linked immunosorbent assay and indirect immunoperoxidase assay. We can also perform the laboratory diagnosis by histological examination of taking a biopsy from the rash. We can inoculate it into cell lines, Vero, WI38, HeLa, also in egg inoculation, embryonated egg in the yolk sac or in the animal guinea pig. By polymerase chain reaction, we can detect the 16 sRNA or the outer membrane protein antigen genes. Treatment for rickettsiosis, we can treat it by antibacterial agents like doxycycline, tetracycline group, and chloramphenicol can be used as an alternative. Prevention we have is vector control strategies such as insecticides, control of rodents and other animals, improvement of personal hygiene, and currently no vaccine is available against rickettsial infection. So after the typhus group and the spotted fever group, we have scrub typhus, which is caused by Orientia shushugamushi, formerly which was classified under Rickettsia, but it differs from Rickettsia both genetically and by its cell wall composition. It lacks the lipopolysaccharide layer in the cell wall. What is the naming? The nomenclature comes from scrub, which are the areas where scrub vegetation consisting of low-lying trees and bushes are encountered. So vector, what is the vector? I told you vector previously, it is the larva of the trombiculite mites. Now what is the trombiculite mites of the genus is Leptotrombidium, akamushi in Japan and Deliensis in India. Now larvae called shigers are the only stage that feed on humans, also called shigerosis. Mites can maintain the organisms through trans-ovarian transmission. So these mites, they maintain the organisms. It can pass to the offspring by the trans-ovarian transmission. So what is the clinical manifestation of scrub typhus? It is a triad. Here we again see an eschar. It is a triad of a eschar at the site of the entry of the mite, the larval form, and regional lymphadenopathy and maculopapular rash. Eschar, regional lymphadenopathy and maculopapular rash. This is the triad of features we have in scrub typhus. Non-specifically, there may be fever, headache, myalgia, cough and GI symptoms. What are the complications? Complications may be encephalitis and interstitial pneumonia. So epidemiology of scrub typhus is also very important. There is a zoonotic tetrad which maintains the life cycle. It consists of the trombiculite mites, the larval forms, which is known as the shigers, and the small mammals, which is the field mice, the rats and the shrews, the scrub vegetation or forest, hence name is scrub typhus, and the wet season when mites lay eggs. So there is this world scenario. We have a Shushugamushi triangle. It extends from Japan and Russia in the north to Australia in the south and to Pakistan in the west. In this triangle, there are maximum incidence of cases. In India also there is re-emergence of this infectious disease in the sub-Himalayan belt, Bihar, Rajasthan, Maharashtra, Karnataka, Tamil Nadu and Pondicherry and Kerala. This is the Indian scenario where we have seen a huge uprise or the re-emergence of scrub typhus in the recent years. Also, there has been a rural to urban shift. There are many increasingly reported cases from urban areas, and treatment is by doxycycline, again tetracycline, chloramphenicol or azithromycin can be given alternatively. Laboratory diagnosis, we rely on the antibody detection. We are performing the IgM ELISA, which appear by the end of the first week, and IgG by the end of the second week. In reinfection, IgG antibodies may also be detectable by day 6, with IgM antibody titer being variable. Wheel-Felix reaction, I told you that it is a high titer of heterophile antibodies to the OX-K antigen. OX-K antigen was derived from non-motile strain of Proteus mirabilis. Now, what is the gold standard test for the diagnosis of serological diagnosis of scrub typhus? It is not ELISA, it is not Wheel-Felix test. The serological test which is gold standard is the indirect immunofluorescence antibody assay. So this is the gold standard detection test, and if we perform ELISA, we perform using the 56 kilodalton recombinant major surface protein antigen. But we have to remember, it may be asked as a question like what is the gold standard test? It will be indirect immunofluorescence antibody assay. Also, PCR can be done for detection of the major 56 kilodalton gene, 47 kilodalton gene and the 16S rRNA. Moving on to early causes. So if we go back and remember, we had divided the order Rickettsialis into genus, family Rickettsia and family Anaplasmatici. Anaplasmatici had 4 obligate intracellular organisms: Ehrlichia, Wolbachia, Anaplasma and Neorickettia. So the pathogenic species, they reside in the vertebrate reservoirs and target vacuoles of the hematopoietic cells. The pathogenic species are Ehrlichia schaffnensis, Ehrlichia ewingi, Anaplasma phagocytophyllum and Neorickettia senetsu. Here we are going to mainly discuss about about Ehrlichia schaffnensis, and this is a vector borne transmitted by ticks. Again, it is a tick-borne illness like the spotted fever group, except Neorickettia senetsu, which is by ingestion of fish carrying by the infected flukes. It is mostly seen in the US, except Neorickettia, which is seen in Japan and Malaysia. So clinical manifestations is headache, myalgia, arthalgia, cough, pharyngitis, lymphadenopathy, diarrhea, nausea, vomiting, abdominal pain. So this early causes, they are obligate intracellular. They reside inside the phagosome and multiply to produce 3 bodies: elementary body, initial body and a specific mulberry-like inclusion called morula. So this is again a question which is asked. This morula is seen in neutrophil, which can be detected by GIMSA stain of the peripheral blood film examination. So this again is a question that is being asked. Moving on to Qfever. Qfever is also known as quarry fever. And what is the causative organism? Previously it was classified under Rickettsia. It is Coxiella burnetii. It is an obligate intracellular organism that causes Qfever. For history is that for a long time the causative agent of the disease was unknown, and therefore it was known as query fever or Qfever. Later it was named after the discoverers Cox and Burnett as Coxiella burneti. It is a zoonotic infection. Human, cattle, sheep and goats are the source. So, why we have separated Qfever caused by Coxiella burneti from the Rickettsia? Because it is not transmitted by the vector, arthropod vector. It is by inhalation of the infected dust from soil previously contaminated by urine or feces of diseased animals, and rarely by ingestion of contaminated milk. It is endemic in most Qfever. Coxiella burneti is endemic in most parts of the world except New Zealand and Antarctica, and in India it is reported from Rajasthan, Punjab, Haryana and Delhi. Clinical manifestation: the incubation period ranges from 3 to 30 days. There is acute cases, acute Qfever characterized by hepatitis, interstitial pneumonia, fever, central nervous system involvement, pericarditis or myocarditis. And chronic Qfever develops months to 2-3 years later in 5% of cases and presents as endocarditis in the other cases in chronic Qfever. Laboratory diagnosis: it is a small pleomorphic gram-negative coccobacilli, and therefore it is extremely fastidious, and it is isolated from the buffy coat blood samples or tissue specimen by shell vial technique, for which we will require a biosafety level 3 laboratory. The method of choice is the antibody detection for the diagnosis, which is again indirect immunofluorescence assay, and we can also do the molecular method to detect the Coxiella burneti DNA in tissue specimen by PCR. Here also the drug of choice is doxycycline, and in chronic Qfever we can add hydroxychloroquine to alkalinize the phagolysosome and to render doxycycline bactericidal against the organism. The vaccine: there is a vaccine that has been used to prevent, which is licensed, known as wholesale vaccine Qvax, licensed in Australia, and it is recommended for occupationally exposed workers. Also, we require to practice good animal husbandry, and we can pasteurization of milk is performed because it can be spread from contaminated milk by flash method, which is 72 heating at 72 degrees Celsius for 20 seconds, followed by rapid cooling to 13 degrees Celsius or lower. So, next we have moved to Bartonosis. What is Bartonosis? It is fastidious facultative. What is Bartonosis? So, Bartonosis, it consists of 3 important human pathogen: Bartonella, Hensley, Quintana and basiliformis. So, these are fastidious facultative intracellular gram-negative bacteria, ability to invade mammalian cells and RBCs. So, coming to Bartonella Hensley, it is transmitted by exposure to cats. So, it is transmitted by scratch or bite of the cat. Therefore, the name of the illness is also known as cat scratch disease. It will lead to lymphadenopathy, regional and painless papule at the site of the scratch. This is the first clinical manifestation, cat scratch disease, first disease. And then we have bacillary angiomatosis, which is neovascular lesions involving the skin and the other organs. And we have bacillary paliosis, which is angioproliferative disorder involving the liver and the spleen. Bartonella quintana, that was Bartonella Hensley. Now Bartonella quintana, it is transmitted by louse. It was also seen in World War I in the soldiers that who would lie in the trench, and therefore they would present with a fever which lasts for 5 days, and then again repeat. Subsequent episode occurred every 5 days. Therefore, the name fell as 5 days fever. So this is a mild febrile illness. Trench fever may also lead to bacillary angiomatosis, chronic bacteremia and endocarditis. Bartonella basiliformis, which is the third species, is transmitted by vector sandfly Lutzomia. It produces a biphasic disease. One is or known as Oriya fever or carrion's disease, and Verruga periona, and others like bacteremia and endocarditis. So we have next the laboratory diagnosis of Bartonella infection, where we can use the blood, the lymph node, or the skin biopsies. In the microscopy, we can perform a special stain which is known as Warthin starry silver nitrate stain and immunofluorescence stain, where we can detect the Bartonella hensleyi from lymph nodes. So this may come as a question. And culture, it can be grown like other rickettsia. They are very fastidious, cannot be grown in cell-free culture media. But Bartonella can be grown in blood agar at 37 degrees Celsius, but with a prolonged incubation period of 12 to 15 days. Antibody can be used like antibody detection, like by indirect immunofluorescence assay and enzyme immunoassay. They detect antibodies, and PCR can also be used to amplify the genes against 16S rRNA. Treatment: cat scratch disease. For extensive lymphadenopathy, we can give azithromycin, bacillary angiomatosis, erythromycin or doxycycline for a period of 3 months. Now we are going to discuss few multiple choice questions. So let us recapitulate what we have learned till now. All of the following rickettsia belong to spotted fever group. There are 4 options provided: Rickettsial rickettsii, Rickettsial conuri, Rickettsial typhi, and Rickettsial acari. So I told you, in the spotted fever group, we had Rocky Mountain spotted fever caused by Rickettsial rickettsii. We had Indian tick typhus, which was caused by Rickettsial conuri, Rickettsial pox caused by Rickettsial acari. So the answer here is Rickettsial typhi, because it causes endemic typhus. Remember the mnemonic ENT. Now, Tunica reaction. It is seen in plague, Haemophilus influenzae, Rickettsial musari, and Rickettsial anthrax. So this is Rickettsial musari answer. I have not discussed it ahead previously. Rickettsial musari is also after the discoverer Neil Moser, who discovered Rickettsial typhi. It is Tunica reaction is seen it is an experiment where we inject intraperitoneally. We inject the antigens of the Rickettsial typhi, as a result of which there will be in the guinea pig, where there will be skin scrotal inflammation and the testis will be pulled into the abdomen. So this is known as the Tunica reaction, which is seen in the typhus group, Indian tick typhus caused by Rickettsial musari. Now, Botanist fever. Botanist fever is the caused by, this is again a one-liner question, Rickettsial japonica, conuri, sebrica, and australis. So, australis, it is seen in Australia, sebrica, and conuri, which causes Indian tick typhus. This also I have not discussed ahead, so I am mentioning this here. Botanist fever is caused by Rickettsial conuri. Now, Ehrlichia schaffnensis is the causative agent of Ehrlichia schaffnensis causes ehrlichiosis, which is human monocytic ehrlichiosis. This is the answer. And modular form is seen. This I have discussed ahead. Modular form is seen in Mycoplasma hominis, Bartonella, Quintana, Chlamydia, and Ehrlichia are the options. The answer here is Ehrlichia, where we have seen that in the neutrophil there are some modular formations seen. So with this we come to the extensive discussion on rickettsial infections. So this is my request that all of you go in details about the topic, because it is very frequently asked and almost always repeated question. I'll thank you for your patient hearing.